The ground beneath the research station trembles slightly as another chunk of permafrost shifts somewhere in the distance. A scientist pulls her thermal gloves tighter and peers into the microscope one more time. What she sees there defies everything she thought she knew about the boundaries between life and death. The sample, extracted from ice that has remained frozen since before the pyramids were built, shows movement. Subtle, almost imperceptible, but undeniably present. This is not supposed to happen. According to every textbook, every scientific principle learned in graduate school, the organisms in this sample should be long dead. They have been locked in temperatures well below freezing for over 30,000 years. Yet here they are, responding to warmth like sleepers stirring at dawn. The Siberian wilderness stretches endlessly beyond the laboratory windows, a landscape so vast and unchanged that time itself seems frozen. But beneath this apparent permanence, something extraordinary has been waiting. The permafrost that covers nearly a quarter of the northern hemisphere is not just frozen ground. It is a vault, a repository of life from ages when the world was utterly different from the one we know today. Ancient air bubbles trapped in ice hold atmosphere from when mammoths walked freely across continents that knew no human footprint. The silence of these frozen depths has preserved conversations between predator and prey that ended abruptly 30 millennia ago. In laboratories scattered across the Arctic, teams of researchers have been making discoveries that challenge our understanding of life itself. Each core sample they extract tells a story that spans millennia. Each fragment of ancient ice contains not just frozen water, but frozen time. The drilling equipment penetrates deeper into layers that have remained undisturbed since the last ice age. Steam rises from the boreholes as ancient air meets modern atmosphere for the first time in thousands of years. But these samples contain more than just air and minerals. They hold organisms that have been waiting in perfect suspension for conditions that might allow them to live again. The first hints of what lay buried in the ice came from routine climate research. Scientists drilling into permafrost to study historical weather patterns began noticing anomalies in their samples. Under powerful microscopes, shapes appeared that looked suspiciously like cellular structures. Chemical signatures suggested biological activity that should have been impossible. Genetic material appeared remarkably well preserved despite its incredible age. What followed was a series of discoveries that would reshape our understanding of survival, adaptation, and the very nature of death itself. In 2014, French researchers successfully revived a virus that had been dormant in Siberian permafrost for over 30,000 years. The pathovirus Sibericum was not just ancient beyond imagination. It was functional, capable of infecting single-celled organisms just as it had done in the distant past. The virus awakened slowly, like a giant stretching after an impossibly long sleep. Under controlled laboratory conditions, it began to replicate, to hunt, to live. This discovery opened floodgates of questions and possibilities. If one ancient virus could be revived, what else might be waiting in the ice? The research teams expanded their efforts, drilling deeper, sampling more extensively. They applied increasingly sophisticated techniques to peer into these frozen time capsules. Meanwhile, climate scientists were documenting changes that added urgency to the research. The permafrost that had remained stable throughout recorded human history was beginning to show signs of instability. Temperature sensors buried deep in the Arctic tundra registered readings that had never been recorded before. Average temperatures in the Arctic were rising at twice the global rate. Ice that had never melted in living memory was starting to soften at the edges. Methane gas that had been trapped for millennia began seeping from the ground in visible plumes. The implications became clear with troubling precision. The natural warming of the planet was beginning to unlock these frozen vaults whether scientists were ready or not. Ancient organisms that had been safely contained in ice were starting to encounter the modern world on their own terms. Without human oversight or preparation, the awakening had already begun. Deep in the Canadian Arctic, researchers made discoveries that expanded the scope of what seemed possible. Embedded in ice cores extracted from regions that had been frozen since the last glacial period, they found fragments of much larger life forms. Hair samples from woolly mammoths contain cellular material so well preserved that individual DNA strands could be examined. The mammoth hair still held the scent of ancient grasses that had grown in meadows now buried beneath miles of ice. Genetic sequencing revealed details about these ancient giants that had never been known from fossil evidence alone. But the mammoth discoveries, while spectacular, represented only the most visible aspect of a much more complex phenomenon. For every fragment of mammoth hair, there were millions of microscopic organisms. Each one was a potential window into life during periods that remained largely mysterious to modern science. Some of these organisms had evolved to survive conditions that were radically different from anything existing on Earth today. They had adapted to atmospheric compositions that would be toxic to contemporary life. They had developed metabolisms that could function in temperatures that would freeze modern blood. The permafrost revealed itself to be a three-dimensional library, with different layers preserving different chapters of Earth's biological history. The deepest samples contained organisms from periods so ancient that the continents themselves had been arranged differently. Intermediate layers held life forms from various ice ages, each one adapted to the specific conditions of its time. The most recent layers contained organisms that had been contemporary with early human civilizations. They offered potential insights into how life had adapted to the climate changes that occurred during humanity's rise. Indigenous communities whose ancestors had lived alongside these frozen landscapes for thousands of years watched the research with interest. Their oral histories contained stories of sleeping powers beneath the ice. Tales of times when the earth would warm and ancient things would return to the surface. These were not scientific observations in the modern sense, but they carried an understanding of cycles that operated on timescales far longer than individual human lifespans. The elders spoke of dreams that came from the deep cold, visions of creatures that had walked the earth before humans learned to make fire. As research techniques improved, the catalog of preserved life continued to expand in ways that challenged conventional biological categories. Bacteria that could survive radiation levels that would sterilize most known environments emerged from samples thousands of years old. Viruses that seemed to bridge the gap between living and non-living matter defied every attempt at classification. Fungi that had developed metabolic processes adapted to atmospheric conditions that no longer existed anywhere on Earth's surface. Some specimens appeared to photosynthesize using light wavelengths that had been filtered through ancient ice for millennia. Others had developed symbiotic relationships with minerals that had been crushed and reformed by the immense pressure of glacial movement. Some of the most intriguing discoveries were organisms that appeared to have been preserved in the middle of complex ecological relationships. Entire food webs had been flash frozen during rapid climate transitions. Predator-prey relationships from 30,000 years ago could be observed in perfect detail. The stomach contents of ancient insects revealed the pollination patterns of flowers that had been extinct for millennia. Parasites still clung to host organisms, waiting for the warmth that would allow them to complete life cycles that had been interrupted by sudden freezing. The research revealed that the boundaries between life and death were far more fluid than previously understood. Many of the preserved organisms existed in states that defied simple categorization. They were not alive in any conventional sense, but neither were they completely dead. They were suspended, waiting, holding patterns of biological information that could potentially be reactivated under the right conditions. Their cellular structures remained intact, as if they had simply paused in the middle of essential life processes. Not far from where the initial discoveries were made, other teams were making findings that added new dimensions to an already complex picture. Ancient pathogens, perfectly preserved and potentially viable, represented threats that modern immune systems had never encountered. Diseases that had last been active before the development of agriculture could theoretically become active again. In a world populated by billions of people with no natural resistance to their effects, such pathogens could spread without the natural barriers that had contained them in the past. But the potential dangers extended beyond biological threats. The melting permafrost was also releasing vast quantities of greenhouse gases that had been trapped for millennia. Methane bubbles rose through the softening ice like champagne through glass. Carbon dioxide that had been locked away since before human civilization began entering the atmosphere. Once released, these gases would contribute to further warming, which would cause more permafrost to melt. This would create a feedback loop that could accelerate beyond human ability to control or predict. The pace of discovery accelerated as research funding increased and technology improved. Advanced genetic sequencing allowed researchers to examine preserved DNA at unprecedented levels of detail. Electron microscopy revealed cellular structures that had remained intact for periods that spanned multiple ice ages. Chemical analysis uncovered metabolic processes that had operated under atmospheric conditions completely different from those existing today. Some organisms had evolved to process nutrients that were abundant during ice ages but rare in the modern world. Others had developed defense mechanisms against environmental stresses that no longer existed. Each new discovery raised questions that multiplied faster than answers could be found. If these organisms had survived conditions similar to those that might exist on other planets, what could they teach us about the possibility of life elsewhere in the universe? If they had adapted to dramatic climate changes in the past, what insights might they offer for survival during the changes that Earth was currently experiencing? Could their genetic material hold keys to medical breakthroughs that would benefit contemporary humanity? The research teams began to realize that they were not just studying ancient life, but participating in a process that had been operating for far longer than human civilization. The ice ages had come and gone multiple times throughout Earth's history. Each one had preserved samples of life from different periods and then released them when conditions changed. Humanity was witnessing not an unprecedented event, but the latest iteration of a cycle that had been operating for millions of years. The Earth had done this before, many times, and would likely do it again. The technology required to study these discoveries continued to evolve in response to the unique challenges they presented. Many samples deteriorated rapidly once exposed to modern atmospheric conditions. The oxygen levels that contemporary life depended on proved toxic to some ancient organisms. Others proved impossible to revive despite perfect preservation. Their cellular machinery had been designed for worlds that no longer existed. The window for understanding these frozen archives was limited and closing as climate conditions continued to change at accelerating rates. Some research teams focused on preservation, developing techniques to maintain samples in states as close as possible to their original frozen conditions. Specialized freezers recreated the exact temperature and pressure conditions of deep permafrost. Others concentrated on revival, attempting to reactivate dormant organisms under carefully controlled laboratory conditions. They gradually adjusted temperature, light, and atmospheric composition to ease ancient life back into activity. Still others pursued genetic analysis, extracting and sequencing DNA to understand evolutionary relationships that spanned incredible periods of time. The discoveries continued to challenge fundamental assumptions about the nature of life itself. Some organisms appeared to have survived multiple freeze-thaw cycles over thousands of years. This suggested that they had mechanisms for entering and exiting dormancy that were far more sophisticated than anything known in contemporary biology. Others seemed to have adapted to gradual changes in their frozen environment. They had evolved slowly even while technically dormant, responding to subtle shifts in temperature and chemical composition. As the research progressed, it became apparent that the frozen giants were not limited to biological organisms. The ice also preserved atmospheric samples that provided windows into environmental conditions during Earth's distant past. Ancient air bubbles contained greenhouse gas concentrations from periods before human influence. The ratios of oxygen to carbon dioxide revealed details about plant life that had thrived and died before the first human settlements. Mineral formations revealed details about volcanic activity, meteor impacts, and other geological events that had shaped the planet's development. Dust particles from ancient storms carried pollen from forests that had covered regions now buried beneath ice sheets. The implications for understanding current environmental challenges became increasingly clear. The preserved organisms had survived dramatic climate changes that dwarfed anything in recorded human history. Their adaptations might provide crucial insights for helping contemporary species survive the changes that were currently underway. Their genetic material might contain solutions to problems that humanity had not yet learned to solve. Some ancient bacteria could break down compounds that were toxic to modern life. Others could synthesize nutrients from raw materials that contemporary organisms could not process. Meanwhile, the melting continued at rates that surprised even the most pessimistic climate projections. Permafrost that had been stable for millennia was softening at depths that had never before experienced temperatures above freezing. Research stations built on what had seemed permanently frozen ground began to shift and settle as their foundations softened. Ancient organisms that had been safely contained were beginning to encounter modern ecosystems whether scientists were prepared for the integration or not. Rivers that had run clear for centuries began to turn brown with organic matter released from thawing permafrost. The ethical questions multiplied as quickly as the scientific ones. Did humanity have the right to deliberately revive organisms that had been dormant for thousands of years? What responsibilities came with the knowledge that climate change was naturally releasing these ancient life forms? How could the potential benefits of studying preserved organisms be balanced against the risks of accidentally releasing dangerous pathogens? Research protocols became increasingly sophisticated as the scope of potential consequences became clearer. Containment procedures that had been developed for handling contemporary dangerous organisms were adapted for dealing with ancient ones. But how do you contain something whose behavior cannot be predicted based on modern analogies? International cooperation increased as governments recognized that the implications of this research extended far beyond national boundaries. Ancient viruses released in Siberia could potentially affect ecosystems on every continent. Greenhouse gases released from Canadian permafrost could influence weather patterns across the globe. The catalog of discoveries continued to expand in ways that defied prediction. Ancient plants contained genetic information that could potentially help modern crops adapt to changing climate conditions. Seeds that had been frozen for millennia retained the ability to germinate when carefully thawed. Preserved microorganisms revealed metabolic processes that could have applications in biotechnology, medicine, and environmental remediation. Some could produce antibiotics that were effective against modern drug-resistant bacteria. Frozen animal tissues provided insights into evolutionary relationships that filled gaps in the fossil record. Blood cells preserved in ice revealed details about immune systems that had evolved to handle environmental stresses that no longer existed. But perhaps the most profound discoveries were the ones that challenged basic assumptions about the relationship between past and present. The organisms emerging from the ice were not simply historical curiosities. They were potential participants in contemporary ecosystems. They were carriers of genetic information that could influence the future evolution of life on Earth. Some might prove beneficial, contributing genetic diversity that could help modern species adapt to changing conditions. Others might prove disruptive, competing with existing species or introducing diseases for which no treatments existed. The ancient predators might find that their traditional prey had evolved beyond recognition. The ancient prey might discover that their defense mechanisms were useless against modern predators. In research stations across the Arctic, scientists worked around the clock to catalog and understand these discoveries before they were lost forever. The race was not just against time, but against the accelerating pace of climate change itself. Each season brought warmer temperatures and more melting. Each year revealed new organisms and new questions. The microscopes revealed cellular structures that had remained perfectly intact for thousands of years. DNA strands uncoiled under careful laboratory conditions, revealing genetic codes that had been written when the world was young. Proteins that had been folded into dormant configurations began to unfold and resume their ancient functions. The scientist adjusts the microscope focus one more time, watching as the ancient organism responds to carefully controlled changes in temperature and light. After 30,000 years of perfect stillness, it moves with purpose that suggests memory. Intelligence that transcends simple biological programming. Adaptation that had been waiting for exactly these conditions to resume. The frozen giant is awakening, and no one can predict what it will do when it finally opens its eyes to a world that has changed beyond recognition. The ice holds its secrets close, but it cannot hold them forever. Soon, very soon, we may discover what giants have been sleeping in the frozen depths. Whether the world they wake to will welcome their return or struggle to contain forces that time itself had thought to bury. The ancient world stirs beneath our feet, and its dreams are beginning to seep into our reality. One thawed cell at a time. The permafrost trembles. The giants stretch. And somewhere in the darkness below, something ancient opens its eyes.